Yeah. Trouble on my mind. I got trouble on my mind. Trouble on my mind. So much trouble on my mind. Trouble on my mind. I got trouble on my mind. Trouble on my mind. Yeah. Trouble on my mind. I got trouble on my mind. Trouble on my mind. So much trouble on my mind. Trouble on my mind. I got trouble on my mind. Trouble on my mind. So much trouble on my mind. I got trouble on my mind. Trouble on my mind.
Welcome to Genetics with the Squad! Video we will be covering Gregor Mendel and what he discovered, types of dominance, meiosis, and inheritance between the two organisms. I'm going to pass it to Matt.
Matt! Thank you, Keo. Gregor Mendel is often called the father of genetics. He used pea plants to study the 7 basic characteristics of life. To explain these characteristics, he came up with 3 laws. The law of segregation is where homologous genes separate during meiosis. The next law is the law of independent assortment. It's when traits are inherited separately from others. The third one is the rule of dominance, and it states that the dominant allele will show up over the recessive one. Since we're on the subject, we're going to dominant and recessive traits. A dominant trait means that it's the trait that will show up. The recessive trait is the one that is hidden unless there are 2 recessive traits. This brings us to our next subject, pedigrees. A pedigree is a family tree in a sense that it shows the genetics and traits that are passed from generation to generation. The traits that you can use are phenotype and genotype, and the phenotype is the physical characteristics of someone, and genotype is the DNA code of the person.
Incest if you're adopted. Genetics.
Now Ethan has a dominant gene for tallness, capital T. Keon here has a recessive gene for shortness. And if these two both cross genes and were heterozygous, then it would be a 75% chance that their baby would be tall. 25% it would be short. Thank you for this example of genetics.
Sometimes I lick my dog. Genetics.
This is where a dominant allele covers recessive. Incomplete dominance is where the dominant allele does not completely cover the recessive. It's pretty much a mixture of the dominant and recessive. Codominance is where it shares dominance. Both show up. The dominant and recessive both show. No mixture. Multiple alleles is where there's more than 2 forms of the trait. You usually see multiple alleles dealing with blood. Sex-linked traits are inherited on X chromosome. To write the genotypes for sex, linked traits, you must include the sex chromosome. Sometimes I like my meat, and sometimes
Non-disjunction is when chromosomes do not separate that well during meiosis.
Oh yeah?
Many female chromosomes have these traits you need to make and produce kids. A pedigree is an ancestral line or chart depicting the lineage or descent of an individual. To shape my body for genetics.
Haploid is a cell with half the number of regular chromosomes. It's made in the process of meiosis. The cells with a full set of chromosomes are called diploids. Diploids each contain 1 haploid from each parent. The cells from each parent are called gametes. They are sex cells. When these sex cells join, a process called fertilization occurs and forms a zygote, a fertilized egg.
I lick my pimples. Genetics. Sometimes I wake up in the middle of the night just to eat mayonnaise. Genetics.
Meiosis 1 has 2 main purposes. It is the reduction division, so it reduces the number of chromosomes in half, making the daughter cells haploids. And it is during meiosis 1 that most of the genetic recombination occurs. Prophase 1. Just like in mitosis, during prophase, DNA condensation occurs. The nuclear envelope and nucleoli disappear, and the spindle starts to form. The big difference is what is going on with the chromosomes themselves. As DNA condensation proceeds and the chromosomes first become visible, they are visible as tetrads, so tetrads become visible during prophase. Metaphase 1. Tetrads line up at the equator. The spindle has completely formed. It is during prophase 1 and metaphase 1 that genetic recombination is occurring. Take a look at the genetic recombination page to find out about how that happens here. Keep in mind that if it only happens when there are tetrads, so as soon as anaphase 1 gets going, genetic recombination is over. Anaphase 1. Tetrads pull apart and chromosomes with 2 chromatids move toward the poles. Telophase 1. Chromosomes with 2 chromatids decondense and a nuclear envelope reforms around them. Each nucleus is now haploid. Keep in mind that this is not the number of chromatids per chromosome that determine whether a cell is a diploid or haploid, but it is the number of chromosomes and whether they are a pair that determines this. Meiosis 2. The purpose is at the end of meiosis 1, each chromosome still has 2 chromatids. That is double the amount of DNA that a cell should have. So the entire reason to go through meiosis 2 is to reduce the amount of DNA back to normal. Basically, to split the chromosome so that each daughter cell has only 1 chromatid per chromosome. Now, prophase 2. Chromosomes with 2 chromatids become visible as they condense in the nuclear envelope, and the nucleoli disappear and the spindle is forming. Metaphase 2. Chromosomes with chromatids line up at the equator. The spindle is fully formed, although genetic recombination primarily occurs during meiosis 1. The way the chromosomes line up during metaphase 2 can also help to make unique daughter cells. I mentioned this on the genetic recombination page. Anaphase 2. The chromosomes split so that a chromosome with only 1 chromatid heads toward each pole. Telophase 2. Chromosomes with only 1 chromatid decondense and get surrounded by new nuclear envelopes. The 4 daughter cells are now all haploid and have the right amount of DNA. They are ready to develop into sperm or eggs now. Pedigrees are charts that show how a certain trait is passed through families. It can be used to predict the possibility of offspring having the same trait. Punnett squares. Pedigrees are used to determine the mode of inheritance, autosomal dominant, autosomal recessive, and sex length of genetic diseases. Basically, the squares are the males, the circles are the females, and whatever one is shaded in is the one that is infected.
Inheritance between 2 organisms. I screwed up. I forgot to say and right there.
The dominant allele shows up over the recessive one. Incomplete dominance is where the dominant